everyone. Thanks for being here. My name is Camila. I'm a final year PhD student at the University of Helsinki under the supervision of NEE, and today I'll be talking about factors driving the microbiota composition in an insect community. The long-term coevolutionary history between insects and their associated microbial communities have played a significant role in the diversity and evolution of insect-symbiont interactions. And one famous case as example for this is this mirrored phylogeny of Cassidine beetles and Stamira bacteria. Stamira bacteria has been shown to provide or to help those beetles to digest leaves. But besides this function, the microbiota can also help those insects with several other functions. For instance, with reproduction, development, and survival time, but also protection against pathogens. The microbiota composition, and for composition here, I mean the type of microbe and its abundance, can vary within and among insects or hosts. And abiotic, biotic, and neutral factors have been indicated as drivers of such variation. And to understand better this type of variation, we can think of a hypothetical environment with a hypothetical host on it. And this host may also have some endosymbionts that co-evolved with it. So, for example, as the same case of Cassidine beetles and their Stamira bacteria. But the host can also acquire bacteria from the environment. And thus, the environment with different conditions and can offer different pools of microbes for that particular host. And for conditions here, we can think about, for instance, seasonality or habitat disturbance. Then we have the following gap. We actually don't understand what's the relative importance of those abiotic factors and these biotic factors. And biotic factors here, we can think about this coevolution of insects and their symbionts, how these different factors actually play in the composition of those microbiota. But then to understand this is quite important to actually have a better overview of the insect symbiosis. A good example or system to look forward into this gap is the insect community in the Åland Islands. Åland Islands is an archipelago located southwest Finland, and in there we have a huge network of about 4,000 meadow sites that are populated with Plantago lanceolata. This site has been used for several long-term studies, and this is quite interesting because we already have provided several information on those sites. And one study of 2019 classified everything that is present surrounding those sites. And based on this description, we can actually categorize those sites as being surrounded by human disturbed areas or natural areas. Those human disturbed areas have, for instance, agricultural fields or buildings, while the natural areas are surrounded by forests or rocks. So based on this, we can go back to our gap and ask the following question. How do the diversity, how do the seasonality, habitat disturbance, and host species affect the Plantago lanceolata insect community and their associated microbes in the Åland system? So let's first look at the insect community in that particular system. So for this, we visited 30 sites in the Åland Islands, and those sites were either surrounded by natural areas, here in dark green, or by human areas in pink. And we visit this throughout the spring and summer of 2021, comprising the months of May, June, and July. So we collected insects that were found in Plantago lanceolata, either on their leaves, their stem, or flowers. And to standardize our collections, we set up 1 square meter quadrats, and the quadrats had to be covered with at least 50% of this area by Plantago lanceolata. In the end, we collected about 2,000 insect samples, and they were grouped in 250 morphotypes. So this is our first result regarding the insect diversity in those communities. So each point here is one patch. They are colored by month of collection. In purple, we have May. In orange, we have June. And in green, we have July. And we see a nice aggregation of those points depending on the month that they were collected. This indicates us that there is a huge effect of seasonality in that particular insect community. However, when we paint those points by the habitat disturbance. Here we have pink as human disturbed area or green as natural area. We see a huge overlap of those points, so we didn't actually find any effect of the habitat disturbance in the insect composition. And this can be explained actually by what we observed in the field. So when we go to the field, we see that in May, the plants are very small because this winter just ended, the snow just melted. But then in June and July, we have a huge peak of flowers. And it's interesting that each time point here can actually attract a different insect community. And on the other hand, the surrounding areas doesn't affect much. And this can be explained by the fact that although the surrounding area quality varies, the quality of the site doesn't vary that much. And then the surrounding area may not offer a huge obstacle for the insects to disperse among those sites. But okay, we can now look at the microbial composition and how it is affected then by the insect species, by seasonality, and habitat disturbance. For this, we went back to our 250 morphotypes, and we selected the 8 most common and abundant. And then we ended up with 3 Hemiptera, 3 Hymenoptera, 1 Lepidoptera, and 1 Coleoptera species. And then we did a full-body DNA extraction. We followed all the steps necessary for the next generation sequencing, and we used 2 markers in this step. We used CO1 to confirm the identity of the insect, and we also used 16S to obtain the bacterial ID. And this is our first result for the microbial composition. And here is also a PCOA plot. Each point here represents 1 insect sample, and they are colored by insect species, and the color code is up there, up the plot. And we also see a nice aggregation of those variation in the insect microbial community depending on the insect species. In fact, like the host here can explain about 60% of the variation that we found in the microbiota of those samples. And this can also be observed if we look at the weaving variation of those insects. So here we have a relative abundance plot. Each tiny bar plot here is one species, and the color code represents 1 bacterial strain. So here we use a ZLDU level, which is the same as ASV. And we observe there is actually 1 or 2 most common, very predominant bacteria in each of those species. This is interesting because it shows us that those bacteria have a very long coevolution with some bacteria, such as the case of those capsid inhibitors and Stamera bacteria. But it also shows us a limitation, because then to continue our analysis looking at the effect of habitat disturbance or seasonality, it doesn't make sense to mix it up all those species together, because each host may actually imply different selection pressures in their own bacteria. So for this, we will proceed separating the data per species, and for a matter of time, I will only show you 2 species, which are Formica aquilonia, 1 ant, and 1 beetle that is Mecinus bascorum. And we also have 2 ways of looking at this data. The first one is looking at the full microbiota study, and here we actually didn't find an effect of seasonality for neither the Formica aquilonia or Mecinus bascorum. And also we didn't find an effect of habitat disturbance in the microbiota composition for neither of those species. Another way that we can look at this data is partitioning the data even more. So we can also exclude from the data the most abundant bacteria to see how the rest is actually behaving. And for this, we excluded from the Formica aquilonia 2 strains of Wolbachia. And for the Mecinus bascorum, we excluded 1 Morganellacea, which is Arsenophonus in this case, 1 strain of Wolbachia, and 1 strain of Rickettsia. So here when we tested the effect of seasonality, we didn't find an effect of seasonality for Formica aquilonia for the partial microbiota data, but we find some effect for the Mecinus bascorum. But this was observed mostly in the STAST results, and here the plot is a little bit funny. I will go back to it later. And then when we go back to the habitat disturbance effect, we also didn't find an effect for the partial data for Formica colonia or Micinus pascorum. What the stats results showed us for the partial microbiota data, that there is an effect of seasonality. What we think that it could be happening in this case is that we are sampling 2 different generations in the field. So when we go to the field in June, we see the insects copulating, and this could be the insects that diapause throughout the winter, and they just woke up after the first, the spring. But then when we go back to the field in July, we could actually be sampling these samples that were offspring of the samples from June. And then again, when we see the effect of the habitat disturbance, they were absent for every analysis that we saw, and the explanation for this could be the same that we have for the insect community. That what could matter more in this case is the quality within the site, not surrounding the site. Finally, when we look at the composition of those insects, we also see some strains that are repeating in different insects. So in this case, we have Wolbachia here in dark green, and also Rickettsia in orange, and they appear in different insects. And when we look at the ecology of those insects, here I have a brief summary of it. So we have the caterpillar there is a moth, and is a leaf roller insect, and it feeds on the leaf. The Micinus pascorum, as a larvae, feeds on the seeds of those plants, but as an adult feeds also on the leaf. And the Mesopolopus incultus, which is a parasitoid wasp of that Micinus pascorum. So here we have those 3 insects connected throughout this ecological interaction. And it could be indicated us that actually there is a possible transfer of Wolbachia and Rickettsia throughout these interactions. But of course, this only opens several more questions. One of them is regarding the horse-shaped plot that I showed you. I'm currently looking into correcting those artifacts, and if you have some ideas on how to fix it, I'm happy to listen to it. I would be very happy to know more about life history traits and also phenological traits of those insects to understand better what's going on in that community and how to connect it with what we found in the microbiome information. And finally, about the host transfer, the horizontal transfer that we observed is just a suggestion because we just based this in 2 markers. So only a whole genome thing would actually tell us if there is actually a transfer going on in this case. So just to conclude, I would like to get back my question here. How does seasonality, habitat disturbance, and host species affect the Plantago lanceolata insect community and their microbiota associated? And then we have that insect community composition is mostly influenced by the seasonality. The host community composition is actually very much influenced by the host insect. Actually, 60% of this variation is explained by the host insect that we found. And then depending on the species, we can also see some sort of seasonality variation taking place in here. I would like to thank my group, the fundings, and every type of support that I received to come here today. And of course, I would like to thank you for listening. Thank you.
Hello, everyone. Thank you for being here today. My name is Amundsun. I'm a PhD candidate at the University of Virginia. And today I'm going to talk to you a little bit about a project that I've been working on with my co-author, Jana Santanovics, that I'm very excited to share. So one of the first things to know about pathogens is that transmission is really important for pathogen success. It comprises a huge portion of pathogen fitness. And yet, despite this, we see a wide variety of ways in which pathogens transmit. So, for example, pathogens can transmit as airborne infective stages released during coughing and inhaled. They can transmit through skin-to-skin contact, or be carried by vectors, or be spread during sexual contact. So given this wealth of diversity in transmission routes, it's sometimes difficult to meaningfully categorize these different routes in a way that can help us make sense of what is happening in transmission and what transmission traits are important. So one way that we can do this is by categorizing routes as being density dependent or frequency dependent. So density dependent routes are those that increase transmission when the host population is very dense. So if you think of your chances of getting a cold when you're hiking in a sparsely populated area versus getting a cold at a crowded airport, that's sort of the prototypical example of a density-dependent transmission route. Whereas frequency-dependent transmission routes don't depend upon the density of hosts. And so they transmit just according to how common the disease is in the population. If a large proportion of hosts have the disease, they will transmit more often. But if very few hosts in the population have the disease, they will transmit accordingly. And so this often is a good model for vector-borne disease or for sexually transmitted disease. And we also represent these two different modes in different ways mathematically. So density-dependent transmission, we often model it as being the number of susceptible hosts times the number of infected hosts times some transmission coefficient. So you can see that it's the contact rate between those two categories of host that matters for transmission. Whereas frequency-dependent diseases, we're more interested in how often susceptible hosts come into contact with infected hosts, given that they come into contact with some host in the population. So you can see that it's the number of susceptibles times the proportion of the infection in the population, and here being the total number of hosts in the population. And something that I'm really interested in is what happens when these two routes are active in the same population, in the same host-parasite interaction. And I'm going to refer to this as mixed mode transmission. So you can see here my little schematic. We have susceptible hosts and we have infected hosts, and hosts are moving from the susceptible to the infected class via these two different mechanisms. And this is very interesting to me because mixed mode transmission, mixed mode transmission is actually really common in pathogens that we see in real life. So being frequency dependent or being density dependent, those aren't mutually exclusive transmission modes. And I have a citation here. This is a really interesting review paper where the authors looked at many sexually transmitted and non-sexually transmitted diseases and sort of synthesized that information to draw more general conclusions, and found that most of the diseases, whether they were categorized as sexual or non-sexual, could also transmit via other means. And non-sexual transmission is often density-dependent transmission. So these two things aren't exclusive. And even a small amount of mixed modedness can completely change our expectations for the dynamics of an epidemic. So in a primarily density-dependent disease, for example, adding a small amount of frequency-dependent transmission or vice versa can change our expectations for whether a pathogen becomes endemic in a population or goes extinct, or whether it drives the host population to extinction. So even a tiny bit of mixed modedness really matters. And I'm really interested in why this mixed modedness is so common. We know that transmission is really important for pathogen fitness. So if you have a successful strategy, then why do we see this persistence of diversity even within a single disease as far as transmission route? So my question is whether multiple modes of transmission are better for the pathogen than just one. So the hypothesis that I'm testing is whether mixed mode transmission benefits the pathogen over single mode transmission. And to test this hypothesis, we built an SI model. So we have these two compartments for our hosts. All hosts fit into one compartment or the other. Hosts are either susceptible to the disease or infected with the disease. And they move from the susceptible compartment to the infected compartment through density-dependent transmission and through frequency-dependent transmission. And to figure out whether mixed mode transmission is beneficial to the pathogen, we varied the proportion of each of these two routes that's happening via this investment parameter. So when investment is 0, transmission is completely density dependent. There is no frequency dependent transmission. When investment is 0.5, we have 50-50 density dependent and frequency dependent transmission. And when investment is 1, the disease is completely frequency dependent. Yes, okay. And then to assess whether this has any benefit to the pathogen, whether the mixed modedness has benefit, we assessed the prevalence of the pathogen in the population. So this is just the number of infected individuals in the population over the total. So when prevalence is 0, there are no infected individuals and all hosts are susceptible. When prevalence is 1, every host is infected. And there are some other things happening in the background of this model too. So we have demographics going on. Hosts give birth, they die, and we assume that the disease is virulent, and so infected hosts have a higher mortality than susceptibles, and or a higher sterility, or a lower fecundity is another way to say it. So, but all hosts experience whatever we've decided the consequence of the disease is. Oh, yes, okay. Okay, so I'm going to show you some possible outcomes for this project. So each of these panels will be like one simulation, but all of these simulations are done over a wide range of parameter space. So one possibility is, here we'll have our complete density-dependent disease on the far left there, and it has some prevalence. And then we'll have a completely frequency-dependent disease on the right of this graph, also having some prevalence. And our kind of null expectation for what might happen is that the prevalence of a mixed mode disease is additive, that it has a simple linear relationship between these two things. So if we have just like a straight line here to plot kind of our null expectation of what might happen. So possible things that could happen. We might see that mixed mode disease or mixed mode transmission results in a lower prevalence than either single mode pathogen. So here we would expect that mixed mode disease is selected against. On the other hand, we could have mixed mode disease performing better than one of the single modes, but not better than the other. So in panel B, the intermediate investment disease, i.e. the mixed mode disease, is doing better than the completely density-dependent disease, but not better than the completely frequency-dependent disease. Or we could have something like this, which is the option that I'm really interested in, wherein the mixed mode disease has this kind of peak in prevalence. So the mixed mode disease is performing better than either single mode disease. So what did we actually find? So here again, same kind of plot. These are two representative examples from the simulations that I run. We found that sometimes the mixed mode disease performs worse than either of the single mode disease. And sometimes it performs better than one, but not the other. So it could be higher or lower than expected. So it was clear from these simulations that we didn't see a simple additive relationship. But we never had a maximum prevalence at the intermediate investment. So the mixed mode disease never performed better than the more successful of the single mode diseases. So panel A, example of how mixed mode disease might be selected against. And in panel B, mixed mode disease might be selected for over, in this case, density dependence, but not over frequency dependence. So I went back to the model. We thought about our assumptions for this initial model, and I mentioned that one of our assumptions was that every host has the same fitness consequences regardless of how it gets the disease. This, of course, isn't necessarily what we see in reality. So often the way that a host gets a disease does have an impact upon what kind of fitness consequences it experiences, whether that's the site of infection being tissue specific, or often we think of sexually transmitted diseases which are frequency dependent, often having a cost to fecundity rather than mortality. So I relaxed this assumption on the model, and we divided our infected compartment into two. So now we're keeping track of whether hosts get the disease via density transmission or via frequency-dependent transmission. And we make the infected hosts that got the disease through density dependence have a higher mortality and then a higher sterility for the other class. And now what we found is that, again, we could see mixed modedness have a higher or lower prevalence than our null expectation. But we also found cases like this one in panel C, where the mixed modedness actually has a higher prevalence than either of the single modes alone. So of course, then my next question was, well, what differentiates the circumstances in which we see panel C's sort of result versus this other kind of result in panels A and B? So I iterated over a broad parameter space, and I'm going to show you the two parameters that I think make the most difference in this relationship, which perhaps unsurprisingly are the severity of the fitness consequences. So on the x-axis, this is how likely the disease is to cause mortality in an infected host, and on the y-axis, we have the sterility, so the severity of the frequency-dependent infected hosts. And I found that the lower the disease-induced mortality, but the higher the disease-induced sterility, the more likely we were to see mixed mode disease be more successful than the single mode diseases. So again, might be higher or lower than expected, but this maximum prevalence at an intermediate investment mode is more likely when there's low mortality and high sterility. So this only happens when this fitness consequence differs by transmission. So to sum things up, sorry, I know I'm almost out of time. That's my chime. Yeah, so we found that mixed mode transmission can confer an advantage to the pathogen, but only in certain conditions. And in the future, I'm excited to incorporate some evolutionary dynamics into this ecological model. Thank you so much.
Hi, everybody. Thanks for coming to my talk. I apologize. My voice is, I'm losing my voice a little bit, so hopefully you can all hear me. I'm currently a postdoc with Bruce and All at UC Davis, studying methods for understanding how mutation events affect transmission rates in viral outbreaks. So I think everybody would agree that a key goal of disease surveillance and mitigation is understanding what mutations affect transmission rates through the host population. I'm going to call these, wrong button. I'm going to call these transmission-enhancing mutations, or TEMs for short. There are phylogenetic methods that we can use to understand how particular mutation events affect transmission rates. So for example, we can have, am I close enough to the microphone? Okay. There are methods that let us sort of paint the tree according to what the transmission rate is on those branches as a function of some a priori defined amino acids, for example. But what we lack are methods that let us infer from the genome which mutations are causing changes without any prior knowledge of which mutations are important. So we've developed an explicit method that allows us to essentially scan the genome for TEMs. And that method is based on a stochastic branching process model. And I'm going to walk you through how this model works by showing you what a realization under the model looks like, what a simulation would look like. So this is what a simulation looks like under this process. I'm actually going to jump over here and just speak loudly. So the process begins at some time 0 in the past with a single lineage with a particular nucleotide. So in this case, the process began with one lineage with nucleotide C. It then evolved forward in time, experiencing transmission events. So this represents an infection spreading to a new host. The new host also has a C at the relevant site. There are going to be sampling events where we collect viral genomes. There are going to be mutation events that change the nucleotide state, in this case from C to G. Recovery events, these are actually death events, but in the model they're death events. In reality, what they represent is the host becoming non-infectious. There are different kinds of, there are transmission events that occur at different rates. So here we had a mutation to A, and then a transmission event that occurred to a lineage with nucleotide A. And maybe more importantly, we have some mutations that confer an increased birth rate. So in this simulation, your cell, I'm thinking of RNA viruses, is increasing the transmission rate of the virus through the host population. So our goal is to understand which sites in the genome have an effect like this. So the process is characterized by a few sets of parameters. In this case, we can imagine there being one transmission rate, which is labeled lambda, for each nucleotide. Then there's a recovery rate, mu, sampling rates, which can be informed based on our knowledge of how samples were collected over time, and what we're calling Q, which is just a standard mutation or substitution model that describes how nucleotides change from one nucleotide to another. This is for a single nucleotide, but the reality is there are probably many nucleotides that affect transmission rates. So we want to extend this to a more complicated case where there are multiple. This is how it works when there are two. So instead of this history representing one nucleotide, it now actually represents a combination of nucleotides. So I have two nucleotides that are evolving, and I'm simply enumerating the birth rates, the transmission rates, for each combination of nucleotides. So what I do is I have a few basic parameters. I have this thing called lambda naught, which is sort of the base transmission rate. It's the transmission rate for a pathogen that has no transmission-enhancing mutations. And then there's a separate effect for each transmission-enhancing mutation. And we simply have a multiplicative model that works like this. We enumerate all possible combinations of nucleotides, and then we fill in what the transmission rate is for each of those nucleotides based on what the TEMs are, right? So in this case, is that CC? I can't actually see it from here, I'm nearly blind, but I think that's CC. And it has no TEMs, so it has the standard transmission rate. When there's one TEM, we just multiply by the effect of that TEM. When there are two TEMs, we multiply them together. So it's simply a multiplicative model, and we can do that for all combinations of nucleotides. And we can imagine doing this for 2 or 3 or 4 total TEMs. All we have to do is be able to enumerate all combinations of nucleotides at the sites that change the transmission rate. That's the basic model, but our goal is to infer what the TEMs are. So I'm going to try to explain how that works. So we're going to represent the TEMs with this object M, which is like the TEM model. And it basically just looks like a list of the effects. So in this case, M is just a list of the effects associated with these two nucleotides at positions 1 and 2. An alternative is that there's just a single TEM, or that there are actually no TEMs. A special case is that there are no mutations that would affect the transmission rate across the entire genome. Or a more complicated model that has 3 TEMs and so forth. So we can sort of arbitrarily imagine that there's a whole bunch of different ways that we could enumerate TEMs. TEM models, I should say. So we're doing this in a Bayesian context, so our goal is to estimate the posterior probability distribution of this model object M. There's a whole bunch of stuff here that I'm hopefully going to ignore, so feel free to ignore it with me. But this is proportional to the probability of the tree and the TEMs, given the model and the other model parameters. This is the probability of the sites in the alignment that do not have TEMs. And this is just a standard phylogenetic likelihood function, so I'm not going to go into any detail here. We have to have a prior on this M object, and then we have other priors in the model that aren't particularly relevant. So for the purposes of this talk, I'm just going to focus on these two things on the right that are most important. I'm not going to go into a crazy amount of detail, but this first thing here is the probability of the tree and the TEMs. And unfortunately, this model does not have very straightforward analytical likelihood function. So in order to calculate this probability, we actually have to use some pretty intense numerical calculation machinery. And this is all developed from these state-dependent birth-death models first pioneered by Madison and colleagues. And we've implemented it, my collaborator Xavier and I have implemented it in this TensorPhylo package that does it very quickly. One thing to note here is that the number of sites that have TEMs puts a very strong limit on the runtime. It's not the right way of saying that. The runtime increases very quickly as the number of TEMs increases because we're enumerating all possible combinations of nucleotides. So L is going to be the number of TEMs in the model. The time it takes to compute this probability scales with L to the 4th power. So in practice, this method is currently only working for like 3 or 4 total TEMs. But we're hoping to relax that in the future. The other important part here is the prior on models M. And this gets a bit complicated, but I'm just going to break it down and point out one important feature. So we have a prior on the number of TEMs. And this is important because this is how we make the method actually work by not trying to explore models with more than, for example, 3 TEMs. So we have a prior probability that there are L total TEMs in the model. And that's going to look something like this. We're going to have some prior distribution that says there are between 0 and 3 TEMs. And there's some flexibility in how to specify this prior. But this is what we're starting with now. So we've implemented this in using reversible jump Markov chain Monte Carlo that samples these model objects M in proportion to their posterior probability. And we've implemented it in this R package, which, is that what I'm talking about? Yeah. This R package called ITEM, which stands for Identifying Transmission Enhancing Mutations. It's actually a working title. If anybody has a better idea, I'm all ears. So the output of running this program is going to be, if you're familiar with Bayesian inference, this should be very familiar, but what you get is basically a text file where each row corresponds to one sample from the posterior distribution. And so we're going to have a sample of models that are sampled in proportion to their posterior probability. And you can do things like summarize the marginal posterior probability of particular TEMs. So in this case, we see that the TEM 3U has a posterior probability of 1. Very strong evidence that that particular site/nucleotide confers a transmission enhancing effect. We also can compute the posterior distribution of the effect for particular TEMs. So in this case, this is simulated data. The vertical line is the true value. So it looks like this, in this case at least, the method is estimating the transmission rate effect fairly accurately. So we've assessed the performance of this method using simulation. I'm not going to go into a whole lot of detail, but we simulate data with parameters that are calibrated by our empirical data analyses. We simulate a varying number of samples, so trees with 250 to 1,000 tips. And then we applied the method to the simulated data to get a sense of how powerful the method is and how accurate estimates are. So this is one simulation replicate. The uracil, all the red samples are samples with uracil at a particular site that have a 1.7-fold increase to the transmission rate. And when I use the method on this data set, I'll get a marginal distribution of marginal posterior probabilities for TEMs that look like this. And I can imagine deciding on a critical threshold and saying that any posterior probability above this threshold indicates that this TEM is truly a TEM. In this case, since I've simulated the data, I know which of those are true TEMs. So the first bar is true positive because I know that is a site that confers that is a TEM. These other two bars here that are above the threshold are false positives. So I can compute a false discovery rate, the fraction of positive results that are false Oh, geez. So yeah, when we control the false discovery rate, we can get a 5% false discovery rate and about an 87% power irrespective of sample size. We applied this to some empirical data. So we gathered some data based on a prior study by Obermeyer et al., which characterized the fitness effects for various amino acid replacements. And they also characterized the birth rates, transmission rates of various strains over time. We collected 1,000 samples from this second half of 2020, estimated a tree, applied the method. This is the tree we got, and you may already see that there are certain clades that probably have transmission-enhancing mutations. The results we get show that there are at least 3 transmission-enhancing mutations. The first one that has the 100% posterior probability arose twice. The second one arose up in this big clade that I've indicated at the top. Interestingly, there's another one that has non-zero posterior probability that has a very similar pattern of samples. The one on the left actually arose twice. You see there's another arrow down there. So we think that's why the model is preferring this one on the left. A similar story here. This mutation actually arose 6 or 7 times, but there's one big clade that has that mutation down there. This other stack of TEMs are only present in that clade, that big clade down there in the right. So again, similar pattern, but the model is preferring the one that has multiple gains as being transmission enhancing. Perhaps interestingly, this clade here is the alpha strain, which became dominant in the early part of 2021. And this mutation on the left actually corresponds to a particular amino acid replacement that is known to increase transmissibility. So the method is agreeing with prior results and also with experimental case studies. With that, I guess I'll thank you, acknowledge my funding and computational resources, and I probably don't have time for questions, but catch me after if you have any. Thank you.